let's go forward. The second talk will be delivered by Professor from Shenzhen, China. So he represents Alpha Mall Science Limited company and Shenzhen Institutes of Advanced Technology, Chinese Academy of Sciences. So please, you are welcome, Dr. Yuan.
Can you see me now?
Yeah, it's perfect. We can
Okay,
see you and we hear you.
I will share my screen. Just wait. Where is it? Okay. Thank you for your invitation. Today, I'm going to talk about advancing modern drug discovery via computational methods. I'm Dr. Shu Guangyuan. So I first going to talk about some background about the modern drug discovery. As we know that the modern drug discovery is a really complicated and long-term tedious project. When we talk about the drug discovery, it can be divided into two different stages. The first one is research, and the second one is development. So the most important part actually is the research part. We also call this is
Hi, Guang, I can't hear you. Hi, Guang, I can't hear you.
It seems that we have some network connections problem now.
Exactly.
Yeah. Dr. Yuan, can you hear us? It's a chairman. We cannot hear you. Yeah, we have some problems with the voice. Dr. Yuan?
Hi, Guang, you may log out, then log in again. To see
Yeah, probably you can reboot Zoom session. Dr. Yuan? Are there still problems or it's only my opinion? There is no voice.
I can't hear Guang's voice. I can't hear. It's their problem.
Yeah, now it looks better. No voice.
Yeah, we see the slides, but no voice, no comments.
Drug discovery and the clinical development. So the whole process will take about more than 10 years, which usually
It's okay now. Sorry.
Yeah. Normally will take more than 1.2 billion US dollars. So it's quite a high risky process. Now, how can we speed up this process and advance the molecule into clinical trial or even to the market in a more efficient way, the computational methods play more and more important roles, especially with the advancing of the artificial intelligence and the computational methods. As we can see from this pipeline, that computational methods can play different roles in the whole drug discovery process, from the target identification to the virtual screening, to the linear optimization, even to identify the preclinical candidate, the PCC molecule. Even there are some startups or some research groups apply computational methods to planning successful clinical trial or recruiting ideal patients or interpreting the data for the clinics. But our group mainly focusing on the preclinical drug discovery, which means that we will perform the computational drug discovery up to the PCC identification. From this diagram, we can see the international big pharmas are really enthusiastic about advancing modern drug discovery, with artificial intelligence or computational methods. The left-hand side diagram shows us the number of scientific publications from those big pharma in the area of computational drug discovery. The right-hand side panels shows us how many activities, those business activities or business collaborations between those big pharma and the computational drug discovery companies. So both diagrams indicated that applying computational methods to modern drug discovery is really a big trend nowadays. So when we're talking about modern drug discovery, we cannot talk about it without the most important drug targets, which is the G protein coupled receptor. G protein coupled receptor is actually the membrane protein. The protein can be divided into membrane protein and solid protein. As from the name, we can imagine that the membrane protein is embedded into the lipid bilayer, whereas the solid protein normally in the cytoplasmic part of the cell. Gpcr, the gene of Gpcr is counted for the 12th genes in the total human body, and about 40% of the marketed drugs are actually targeting at the Gpcr. Almost all the essential human diseases actually are related directly to Gpcr or are related to the Gpcr signaling. According to the 3D-dimensional structural features of Gpcr, it can be divided into 4 different major types, A, B, C, and F. Whereas the family A is the largest family of Gpcr. It is about 80% of the whole Gpcr family. And the right-hand side of this diagram just shows us the number of Gpcr approved by the FDA. And this light blue is the total number of the FDA approved drug molecule. As we can see, that Gpcr accounts for almost 40%, so which is pretty amazing and important drug targets. So for the Gpcr, and we can see that even for the potential or targets of the Gpcr, it also accounts for a huge and large number of proportions. And here's the statistic of the reported drug molecule from different databases. And the small molecule of Gpcr drug discovery it is still the major trends in this area. And then antibody at this moment, there's only one for the G protein coupled receptor drug discovery. From the point of view of structural biology, and nowadays it is the number of the structure number of Gpcr is also increasing dramatically, as we can see in the early, before 2006, there's only one Gpcr crystal structure were resolved, that is the rhodopsin. And after that, it's increasing dramatically. Up to now, there are more than 120 different Gpcrs were resolved. And in our human body, there were more than 820 different Gpcrs. So which means there is about 20% of the less than 20% of Gpcr structures are actually resolved. So still a lot, the most part of the Gpcr is still unresolved. So Besides the traditional experimental methods such as crystallography or the cryo-EM to resolve the Gpcr crystal structure, using computational methods to make prediction for the G protein coupled receptor as well as other membrane protein becomes more and more popular. For instance, for the AlphaFold 2 and the Rosetta Fold are really being widely discussed these days. Both tools claimed that they can accurately to make predictions for all the proteins, and they published the paper in Nature and Science respectively. However, when we considering the facts that in the whole protein database, the PDB database, there are about 190,000 structures were resolved. There's only 4% of the proteins were membrane proteins in all those resolved structures, whereas the solid proteins occupies 91% of all those resolved proteins. As we know that the Rosetta Fold and the AlphaFold are actually artificial intelligence-based algorithm. It is highly depends on the number and the quality of the initial data. So we can see the PDB in the PDB, the most of protein are actually solid protein, and the membrane protein only occupies less than 4%, which is the number is in total about 6,500. So many people are just wondering whether Google's AlphaFold or Rosetta Fold could also make good performance for the membrane protein structure predictions. So here's a paper which was published in JMB in the early 2021. As we can see, this pink cartoon is the experimental structure, whereas the green cartoon is the prediction from the AlphaFold. As we can see, actually they didn't superimpose well. Even the second structure from the AlphaFold predictions actually doesn't in agreement with the experimental structure. And at the beginning of this year, it is February 3rd, and there's several scientists from the American Academy of Science. They published a common paper in Science. They claimed that they don't agree with some author that the protein folding problems has been solved by either AlphaFold or Rosetta Fold. So that is a really criticized for that. And more importantly, that in the April 13 in 2022, there's a Nature paper has been published by some structural biology. Brian Ross, which is a leading scientist in the area of G protein coupled receptor structural biology and pharmacology from the University of Northern California at the Capitol Hill. He also resolved in his laboratory. He resolved dozens of Gpcr structure, which has been not be published. So he says that about half of the time, AlphaFold structures are very good and acceptable, but half of the time they are more or less useless for their purpose. In some instance, he says AlphaFold labels predictions with high confidence, but actually the experimental structures shows which is totally wrong. So that is even more evidence shows that the AlphaFold actually doesn't perform so satisfactory for the membrane protein, especially the G protein coupled receptor structure predictions. So how to validate that? By the end of 2021, there's a Gpcr contest, which is international contest, to make predictions for the G protein coupled receptor, as well as the ligand binding for the related targets. There are 5 different targets were released, were assigned for this Gpcr 2021. And then last month in April, and the organizer just announced the results and how different computational group behaves performs, and how the Google's AlphaFold or Rosetta Fold performs in this global contest. And our team also joined this contest. So here's the procedure that we made for the Gpcr contest for the last year. We first select the templates for the G protein coupled receptor, then make a 3-dimensional alignment between the sequence, between the targeted sequence and the 3-dimensional experimental structure. Then we generated more than 20,000 initial models, and then we also introduced the molecular dynamic simulation to optimize the model. And we also introduced the in-house AI-based molecular docking to evaluate how those potential compounds bind to the specific target. And finally, we selected the model for the evaluation. So here's the most important and the most difficult part, most difficult targets prediction from our group. So we made a prediction for the best model for the kappa opioid receptor. That 5 of our models, we are ranked as the top 2, top 3, top 5, and also one of them as top 12 for this kappa opioid receptor. From the root mean scale deviation between the experimental structure and our model predictions, we can see the RMSD actually is pretty low. For the transmembrane region of our predictions, it is within 0.9 and 1.5 angstrom. So it is highly in agreement with the experimental structure. And we also use our dedicated workflow, made correct predictions for the binding mode and repeat. We could repeat the binding mode from the cryo-EM structure. All the important interactions between the drug molecule and the receptor can be repeated. For instance, the hydrogen bond with this aspartate, with this tyrosine, also with this tyrosine, with this asparagine, we are all repeated from the cryo-EM structure. So that is pretty accurate for that. So we are just wondering how the AlphaFold or Rosetta Fold performs for this GPCR contest. As we can see, this orange one is the prediction from the AlphaFold, and here's from the Rosetta Fold. And all the RMSD is much higher than our predictions. And in our recent project, we are also working on another G protein coupled receptor. As we can see, that is class C Gpcr, which is much larger. And our prediction is just within 3.8 angstrom, whereas the AlphaFold RMSD is over 50 angstrom, and the Rosetta Fold is even closer to 60 angstrom, which is just also astonished us. Of course, making predictions for the 3-dimensional structure of a protein is not the only way of computational methods that we can do. And we also introduce computational methods to make predictions for the new binding site of G protein coupled receptor, which was also validated by the cryo-EM structure. And in 2020, we also found a new binding mode of G protein coupled receptor, which can coordinate with both drug molecule and the GPCR. With this specific binding mode, we just modify one of these atoms from hydrogen into fluorine, and then we increase the EC50 34 folds, and EC50 is up to 18 nanomole, which is pretty potent. So our group had did systematic work for the G protein coupled receptor, from the drug recognition and the drug discovery to the conformation change, to the water channel mechanism proposal, and also to the transmembrane movements. So with this, I would like to thank to all the collaborators in our group, and my time is over, and thank you for your attention.
Thank you very much, Dr. Yuan. Questions? I do not see any questions in the chat in the moment. So I would like to ask you, when you demonstrated your successful results in a competition of GPCR structure prediction from and comparison with experimental structures, what can you say if you compare finally the experimental structure and the best suitable template which was available before you started your simulations? What was the root mean square deviation, for instance? It's just to evaluate how successful will be this technique in case you have no suitable template in the beginning.
Yes. Yeah, thank you for your question. Of course, to select the template from the beginning, it is very important. As I mentioned from the beginning, GPCR actually can be divided into different subtypes, the A, B, C, and F. And in class A, they can even further be divided into other different subtypes. For instance, they have the peptide family A Gpcr. They have hormone family A Gpcr. They also have this olfactory receptor Gpcr. So with this knowledge, we always select the same template from the same subfamily to use them for the sequence alignment and use them as a template for the Gpcr. And then we also introduced the MD simulation to perform to further optimize the initial models. Of course, we don't know which conformation would be the best one that is close to the crying structure, because you don't know how the crying structure looks like. Normally, MD simulation will generate tens of thousands of different conformations. Normally, I will usually restrain the backbone of the Gpcr. That is because during the MD simulation, the initial model might contain some unfavorable contact. If you don't restrain the backbone, the overall conformation of Gpcr of your homology models will be changed. So usually during the MD simulation, I will restrain the backbone for the transmembrane alpha helix region, and then all the flexible, like the loop, we will keep it to be totally free during MD simulation. And finally, we will make a cluster for the information and select the lowest energy conformation for the further study. Yeah.
Okay, thank you very much. We have one more question in the chat, but probably you can answer directly in the chat session. Because we need to
for the previous Raffaelli versus crystal, we make a model, right?
Yeah, we have strict limitations on our time schedule. And thank you again